Hello and welcome to video 400 in this chapter 6 lecture series. What we're going to talk about next is energy conversion. We know that energy is needed to rearrange matter to make you, right? You take in materials from outside your body and then you use energy in the foods you consume to rearrange you. So what we're going to look at next are a couple of organelles central to that energy conversion process. Mitochondria and chloroplasts. Again, every year on the AP exam, they love to get into the relationship between mitochondria and chloroplasts. We'll get into that a little bit more later in the year. But things to know about mitochondria and chloroplasts. They're both semi-autonomous, much like the Catalan region in Spain and parts of France. So they're semi-independent. They sort of do their own thing, right? So they can do this because they contain their own DNA and they can reproduce on their own schedule. They don't have to reproduce when the rest of the cell reproduces. Again, they sort of operate semi-independently within the cell. So they have sort of different ribosomes and their own unique DNA. And what's really cool about this is that when you look at their DNA and ribosomes, they're actually somewhat similar to bacterial DNA and ribosomes. Another really wild feature of mitochondria and chloroplasts is that they're double membrane. They have two sets of membranes. And all this happened because a long, long time ago, one cell invaded another cell. One cell, the prokaryotic cell, invaded another cell. And rather than destroying it, they formed a partnership, a mutualistic symbiotic relationship. So that's why they're double membraned. When one cell invaded another, now they have two sets of membranes. And what these cells could do really efficiently is convert energy. So it made these cells super efficient and effective. And it's been so effective, it's in all of us. So let's look first at chloroplasts. What chloroplasts do, basically speaking, is absorb energy from the sun and use that energy to rearrange carbon into glucose. So they take energy from the sun and convert it into the chemical energy that you see in the bonds of glucose and other carbohydrates. Now, what mitochondria can do is use the energy in glucose to produce ATP. So they take the hydrolysis or the breakdown of glucose to power the dehydration synthesis of ATP. And of course, oxygen is a big component of that. We'll get into that stuff a lot more in the third unit of the year. All right, so now we get into some of the protein-based structures in the cell. First, let's look at the cytoskeleton. The cytoskeleton has different types of proteins. There are these microfilaments, microtubules. They do slightly different things. But the cytoskeletal elements are actually really important to cells. I think sometimes when people learn about cytoskeleton initially, it's like, you're making your cell drawing and you draw some lines and, it supports the cell. Yes, it does support the cell, but it does so much more. Some cells can actually rearrange their cytoskeletons and change their shapes. Later on the semester in the fourth unit, when we get into cell communication, you're actually going to be able to use pheromones, chemicals released from one organism to another, to make some yeast actually change their shape. And you'll be able to see them change their shape under the microscope because they're rearranging their cytoskeleton. The cytoskeleton can also imagine it being like a spider web within a cell. If you pull on one part of the spider web, it affects the other parts of the spider web. You can actually relay mechanical force or tension through a cell to communicate with the nucleus. And probably my favorite feature of the cytoskeleton is that these proteins serve as like monorails and allow for the transport of materials through cells. We know it's watery inside of cells, right? But it's not like everything's just floating around in a little cellular ocean. Materials within the cell, like vesicles, they actually travel along these roads. They travel along these cytoskeleton proteins. And they do that with the help of little motor proteins. So motor proteins grab the vesicles and actually transport them along the cytoskeleton to destinations. So that usually doesn't get drawn in or put in animations. But in reality, stuff is moving in an organized, coordinated way through the cell. So again, we've got a fun little animation to watch for that. Here are the cool things you can do in college, right? These are onion seeds. Little little post-production sound editing. There, you can grow your hair out, do some studying in the lab. It'll be cool. So there's the dividing tissue in the onion root tip. And now they're zooming in on the root hairs. These have increased the surface area. And what you see there is cytoplasmic streaming, the movement of materials through the cytoplasm. So there you go. There you see the cell wall in the background. And then what we'll do is they'll show, there you see the vesicles. They're the glowing green structures. And then the cytoskeleton, there you go. They're like the ropes traveling through. Oh, you just see a little motor protein. So there's another one. Here we go. So there's a motor protein walking along the cytoskeleton. And then you see on the outside of the vesicle, those are the little like molecular tags that sort of like address labels saying where the vesicles need to go. So that's pretty profound, right? But the idea is change the shape, change the function, right? Use some ATP. Yeah, that's the kind of thing you can imagine, right? Okay. Let's stop the chipper music. But the idea is when you add and remove ATP, those motor proteins change their shape. And when they change their shape, they'll, in essence, sort of walk along this monorail system. They'll walk along the cytoskeleton and, again, transport materials hither and thither through the cell. Okay, some other protein structures, the centrioles and the centrosomes. The centrioles, I'm sure you drew those way back when. Please just note for a moment the shape of the centrioles. It looks like little bundles of, is it fusilli? Is that the pasta type that's got the little hollow tube? Bucatini? I don't know. Anyway, it looks like little hollow tubes, right? And centrioles have a role in bundling together, organizing microtubules that are used for cell division. Centrosomes are areas, they're sort of cloudy regions around the centrioles. So if we look closely, here are the little centrioles that look like bundles of sticks. And the centrosome is this cloudy region around the outside. We'll talk about this more when we get into cell division. But what they basically can do is help hold the cell together while proteins are moving the chromosomes around, all that business. Again, one last time. Observe the structure of the centrioles. This will come up later. Okay, now let's look at cilia. Cilia are little hair-like projections. They're short, skinny hair-like projections on the outside of cells, and they can row like oars. And what that does is it moves fluid past cells. So if it's a little motile cell, again, like certain types of bacteria or protists, what they can do, that rowing can propel the cell through fluid. Now in us, we have ciliated cells, but they're stationary. Our ciliated cells like line our respiratory tract, so they sweep mucus up out of your lungs. Our lungs are covered in mucus to trap the junk we breathe in all day. And what the cilia do is sweep that stuff up to the back of your throat, and then you can swallow that mucus and send it down to your stomach, or depending on how you're raised, spit it out, right? So cilia can help clean out our lungs. Earlier, we looked at the ciliated lining, interior lining of the oviduct to sweep the egg toward the uterus. So again, cilia move fluids. Now, what's cool about this is if you look back here and you see the structure of the microtubules used in reproduction, hey, you can repurpose that stuff, make some modifications, and boom, you've got some proteins related to movement in cells. Now, flagella, similar internal structures between flagella and cilia. It's just that cells have fewer, longer flagella. Okay, a lot of times cells will have one flagella or just a few flagella. But again, you've got this basal construction that's identical to a centriole. So you can see how evolution, with evolution, you start off with division of the cells, but then you can sort of repurpose these proteins and rearrange them into these couplets rather than triplets. So you rearrange them into doublets, or what they're referred to as. And then you can create these structures that are motile. So this is just sort of getting into connecting the anatomy to the physiology of this. So microtubules are types of proteins, right? And if you look in the image here, they're arranged in pairs, so it provides some rigidity. Now between them are these X-linked proteins, or X-linking proteins. They basically hold it all together, so it acts as one big unit. And then what's really cool are these dynin proteins. The dynin proteins, they're sort of like the motor proteins. So what they do is they walk up the length of the tube to start to bend it. Here's the way I sort of conceptualize it. Imagine you have like a pool noodle, a foam noodle on a table, and you start to walk your hands up that pool noodle. Well, what's the pool noodle going to do? It's going to bend, right? Well, that's what those dynin proteins do. These doublet proteins are like the pool noodles, and the dynins are like your hands. So your hands walk up, it bends the noodle, and then that causes the undulation or the rowing of cilia or flagella. The AP exam doesn't get into that super deep. It's just, I think, helpful to sort of understand how it works. Okay, that finishes organelles. We're going to conclude in the last video with ways that cells are connected to each other.